In a lab at Columbia University, researchers took single strands of human hair and did something almost nobody had done before. They measured the color along the length of each strand, micron by micron, like reading tree rings. And on some of those strands, they found a section that had turned gray, and then, further along, turned dark again. One documented hair went from pigmented to white and back to pigmented, all in the same continuous strand. It is worth flagging right away that this comes from a small, observational study, so it is best read as a documented case rather than a settled rule. That means the graying had reversed while the hair was still growing out of the scalp. So the question stops being whether it is possible at all and becomes something much more specific and much more interesting. Under exactly what conditions can a gray hair go back to its original color? Gray hair reversal has become one of the most aggressively marketed corners of the longevity world. You have got topical serums, catalase sprays, copper peptides, expensive injectable treatments in clinics, and a whole shelf of supplements all promising to bring your natural color back. The interest is understandable because hair color is one of the most visible markers of aging we have, and unlike a wrinkle, it feels like it should be a chemistry problem we could just fix. There is a genuine, fascinating layer of real science here about how and why hair loses pigment, and then there is a marketing layer built on top of it that has run miles ahead of the evidence. Today, we are going to separate those two layers carefully. We will walk through the biology of what is actually happening inside a graying follicle, look at what the human data honestly shows, and figure out where the realistic line sits between hope and hype. Let's start with the cells that do the coloring. Inside each hair follicle sit melanocytes, pigment-producing cells that manufacture melanin and hand it off to the growing hair. Behind them is a reserve team called melanocyte stem cells. These stem cells act as a backup supply that steps in to replace melanocytes as they wear out. Think of it like a paint factory with a crew of painters and a bench of trainees waiting to take over. As long as the bench stays full, the factory keeps producing color. When the bench empties out, production starts to fail, and that failure shows up as gray. So, what empties the bench and damages the painters? A big part of the story is oxidative stress. Over time, reactive oxygen species, unstable molecules that damage cellular machinery, accumulate faster than the cell can clear them. One specific culprit is hydrogen peroxide. It builds up naturally inside hair follicles, and in youth, an enzyme called catalase breaks it down efficiently. As we age, catalase activity declines, the peroxide lingers, and it essentially bleaches the hair from the inside while also damaging the pigment cells themselves. The peroxide essentially bleaches the hair from the inside while also damaging the pigment cells themselves. Mitochondria play a surprising role too. These are the energy generators of the cell. When they malfunction, they produce less energy and more oxidative byproducts. But when researchers ran a proteomics analysis, the gray hairs actually contained more mitochondrial proteins, not fewer. That suggests the cells were straining to compensate. They were ramping up machinery in a struggling system, which paints a picture of energy allocation gone wrong, rather than a clean power failure. This is a fascinating observation. Not a lever you can pull, nobody has turned it into a validated intervention. Then there is the stress pathway, and this is where it gets specific. When the sympathetic nervous system activates your fight-or-flight response, it releases norepinephrine. In animal research, that surge of norepinephrine appeared to drive the melanocyte stem cells to activate all at once and burn out, permanently depleting that reserve bench. Stress hormones like cortisol also raise energy expenditure. Pigmentation is a non-essential luxury the body is willing to sacrifice when resources are being pulled elsewhere. And copper matters here as a raw material because it is an essential cofactor for tyrosinase, the enzyme that actually builds melanin. Copper is an essential cofactor for tyrosinase, the enzyme that actually builds melanin. Across all of these, the mitochondrial findings and the stress pathways alike hold a clear line in your head. This is a description of what seems to happen biologically, largely in animal and lab work, not a menu of validated, targetable levers you can currently pull to reverse gray. So, how much of this translates into a hair on your head going back to its natural color? Here is where I want to be precise, because the honest answer is narrower than the marketing implies. The strongest human evidence comes from that Columbia work mapping color along individual strands. In some people, hairs that had gone gray regained pigment, and the researchers were able to line up those color changes against the person's life events. In one striking case, a roughly 2 centimeter white segment of hair mapped almost exactly onto a documented multi-month stretch of intense personal stress. When the stress resolved, color returned. One hair reportedly regained full pigment in about a week after its stressor lifted. This is a genuinely remarkable finding, but it is also a small observational study, so these should be read as cases documented in this particular work, rather than settled fact, and they point to a very specific scenario: stress-induced graying in younger people whose melanocytes and stem cells were still intact and simply suppressed rather than destroyed. Researchers have proposed a threshold window idea backed by mathematical modeling. I want to be clear right away that this is a theoretical construct, not a clinically validated one, and it does not hand you a concrete, actionable timeline to plan around. The idea suggests there is a period during which a graying hair can still recover, before the pigment cells cross a point of no return and become permanently depleted. Beyond that threshold, removing the stress does nothing, because there are no functional cells left to restart. This window concept remains theoretical and has not been clinically validated, so treat it as a useful framework, not a proven timeline you can count down. Beyond that threshold, removing the stress does nothing, because there are no functional cells left to restart. This window concept remains theoretical and has not been clinically validated, so treat it as a useful framework, not a proven timeline you can count down. On the intervention side, there are scattered signals but nothing solid. Certain cancer immunotherapy trials incidentally produced hair darkening by stimulating melanin production, but that came out of an aggressive drug context that does not generalize to healthy people wanting their color back. Minoxidil and stimulation of the mTOR pathway have shown some potential for darkening follicles in early studies, but that is preliminary and not a standard treatment. PRF and PRP injections have also shown inconsistent results. PRF and PRP injections combined with stem cells have produced darkening in some patients, but inconsistently. An A-cell is marketed for pigmentation benefits while lacking direct evidence for reversing gray specifically. So the realistic verdict is this: reversal is biologically possible, but only in a narrow lane. Recent, stress-driven graying in someone still inside that window. Age-related graying in older adults is essentially irreversible with current science. Given all that, what can you actually do that is grounded rather than wishful? The most defensible move is to address the inputs that pigment cells demonstrably need and the stressors that demonstrably suppress them. Start with nutrient status, because deficiency-related graying is one of the few genuinely correctable causes. Low levels of B12, copper, iron, zinc, folate, and vitamin D have all been associated with premature graying. The practical step is not to megadose blindly, it is to actually measure, and to do that through a doctor. Ask your doctor for blood work covering these, and let a physician guide correcting only what is genuinely low. Copper and iron in particular are harmful in excess. Both the testing and any correction should go through a doctor rather than being something you manage on your own. Testing before supplementing is not optional. Second, take the stress pathway seriously as biology, not as a wellness cliché. The norepinephrine and cortisol mechanisms we talked about are the most reversible part of this whole picture. If you are in a younger age bracket and noticed recent graying lining up with a rough patch, genuinely reducing that chronic stress load through sleep and recovery is the intervention with the most credible mechanistic support. If you want to track whether anything is working, the strand is your best instrument. Because hair grows at a fairly steady rate, a hair that changes color partway along its length gives you a rough timeline you can map. On topicals and supplements marketed specifically for reversal, catalase sprays, copper peptides, keep your expectations calibrated to the evidence, which is thin. They are built on real mechanisms but lack convincing human trials. This information is for educational purposes only. Always consult your doctor before making any changes to your diet, supplements, or medication. The biggest real risk is not the graying, it is the correction. Copper and iron are toxic in excess and can accumulate quietly, so supplementing them without confirmed deficiency is genuinely dangerous. Be cautious with expensive clinic packages promising pigment restoration. What I find genuinely compelling about this whole area is not the promise of turning back the clock. It is that a single hair can act as a physical recording of your stress over months, and that in the right window, the body can quietly undo some of it. That tells us pigment loss is not always a one-way door, it just closes faster than most of the marketing wants to admit. If this kind of careful walk through the evidence is why you watch this channel, a like genuinely helps it reach others. Subscribing keeps these deep dives coming. I am curious about one thing in particular. Have you ever spotted a single strand of your own hair that was two different colors along its length? Describe what you saw in the comments.